Okay, so we're going to prepare a slide, a specimen in a slide. So to do that, for our experiment, we need to prepare, of course, a clean glass slide, a cover slip, and the specimen that we have right now is letter E. So to begin with that, we need to place our specimen on a clean slide or glass slide properly. Then to hold the specimen, we need to put, or to mount the specimen, we need to put a bit of water. By putting a bit of water on the slide in order to make the cover slip intact. And this is our slide. In placing the slide in the microscope, we need to properly adjust first slide, and let's check if the microscope is working properly. So here in our setup, I have the slide. I'm gonna put the slide in the stage of the microscope and make sure that the coarse objective is working properly together with the attached coarse adjustment knob on it by moving it downward or upward. Just make sure that the objective is in place